Welcome to this explainer. Today, we're taking raw cellular biology notes and turning them into a fascinating tour of the microscopic city limits. Think of a cell not just as some tiny biological building block, but as a bustling, highly fortified metropolis. We're going to dive into exactly how these cells protect themselves, how they control their borders, and how they manage the vital resources they need to survive.
Here's the roadmap for our Border Patrol tour today: the outer defenses, the gatekeeper, free entry, paying tolls, and finally, heavy cargo.
Kicking things off with Part 1, the outer defenses.
Right in the source notes, you can see the anatomy of the cell envelope is divided into two main physical barriers, the cell wall and the cell membrane. These are the foundational structures of our microscopic city, operating by entirely different sets of rules. To really understand how a cell survives in a chaotic environment, we have to look closely at these two distinct layers of protection and how they function together to keep the inside safe from the outside. First up, the cell wall. It acts like a rigid outer barricade, but there's a major catch. It's exclusively found in plants and fungi. Animal cells completely lack a cell wall. Its primary job is providing structural rigidity, keeping plant cells upright against the elements. It's literally why a towering oak tree can stand hundreds of feet tall without a skeleton. But here's the fascinating part about its border control policy. It's freely permeable. That means, despite being this tough, armor-like wall, it essentially leaves the physical gates wide open for water and other materials to just pass through without much scrutiny.
In contrast, you have the cell membrane, also known as the plasma membrane. Now, this is the universal border patrol present in every single cell on Earth. Structurally, it's constructed from a very specific dynamic mix: lipids, which are fats, and chains of amino acids, better known as proteins. This intricate fat and protein barrier is flexible, it's adaptable, and it's what actually manages the rigorous day-to-day security of the cell.
Move on right along to Part 2, the gatekeeper.
So, the absolute crucial concept from the text is that this membrane is selectively permeable. It only allows the entry or exit of some material. It acts just like a strict VIP bouncer at an exclusive club. It doesn't just let anyone wander in or stumble out. The membrane carefully inspects what's beneficial for the cell to absorb, like nutrients and oxygen, and determines what toxic waste needs to be kicked out. This constant, vigilant sorting is exactly what maintains the perfect internal balance, keeping the cell alive. Now for Part 3, free entry or passive transport.
Passive transport is essentially the free entry lane at our checkpoint. The defining characteristic is that material gets exchanged without using any cellular energy. The cell doesn't have to lift a finger, burn any calories, or spend its precious resources to make this happen. The surrounding environment simply does all the heavy lifting based on natural physical laws.
This free entry lane divides into two distinct subcategories: osmosis and diffusion. While both are elegant forms of passive transport that require absolutely zero energy, there's a very clear biological line between how they operate, the conditions they require, and what exactly they're allowed to move across the border.
Let's break down osmosis first. Osmosis is associated exclusively with a solvent, typically water, and the flow is incredibly specific. It always occurs from an area of low solute concentration to an area of high solute concentration. It's basically trying to dilute the more crowded side. Plus, for osmosis to occur, a selectively permeable membrane is absolutely always required. Without that barrier, you simply do not have osmosis.
Diffusion, on the other hand, handles a much wider variety of traffic. It's associated with both solute and solvent. Notice the directional flow here. It's the exact opposite of osmosis. Diffusion flows purely from high concentration to low concentration, simply spreading out until things are perfectly equalized. And interestingly, a membrane might not even be required. It can take place straight through a membrane, or it can happen just out in the open environment, like a drop of food coloring spreading evenly through a glass of water.
Looking at our source notes side by side, it's crystal clear how these two distinct energy-free transport systems complement each other. You have osmosis specifically moving the vital solvents across strict barriers, and diffusion spreading out the broader traffic wherever there's an imbalance. Together, they keep the cellular border running smoothly without costing the cell a single drop of energy. Which brings us to Part 4, paying tolls, known as active transport. This is where active transport enters the picture, the use of cellular energy to move ions across the membrane. Think of it as paying a biological toll. because the cell sometimes needs to move specific ions against their natural flow, pushing them from an area of low concentration into an area of high concentration. The free entry lane just doesn't work anymore. The cell must actively spend its own energy to literally pump these vital elements through the gate, forcing nature to work in reverse.
And finally, Part 5, heavy cargo, or bulk transport. The notes define bulk transport as the movement of large macromolecules across the membrane. This is the special processing lane for moving truly massive payloads. We aren't talking about tiny sodium ions or simple water molecules slipping through anymore. We're talking about giant biological structures that require a completely different, much more dramatic set of logistics to get across our metaphorical city limits.
This begs a rather funny biological question. How exactly does a microscopic cell go about eating or drinking these massive payloads? I mean, it doesn't have a mouth, so how does it manage this heavy bulk transport?
To handle this, biology gives us specific mechanisms that break down bulk transport into precise directional movements, engulfing things to bring them in and fusing with the wall to push things out. These processes fundamentally reshape the cell membrane itself to accommodate the cargo.
When a cell wants to bring heavy cargo inside, it performs endocytosis, literally engulfing the material. Rather than trying to force a giant object through a tiny, regular gate, the flexible cell membrane reaches out, wraps itself entirely around the massive macromolecule, pinches off, and swallows it whole. The cargo is then safely packaged inside a little biological bubble.
Endocytosis is further divided based on what exactly is being engulfed. If that material is solid, the specific term is phagocytosis, which translates beautifully and literally to cell eating. It's an aggressive, dynamic process used by cells to take in large food particles, or in the case of our immune system, used by white blood cells to hunt down and devour invading bacteria.
On the flip side, if the engulfed material is liquid, it's called pinocytosis. This gives us the highly relatable concept of cell drinking. The cell membrane simply folds inward to create a tiny pocket, scooping up a big gulp of the surrounding fluid and all the dissolved nutrients inside it. So, whether it's chowing down on solid macromolecules or taking huge gulps of fluids, endocytosis covers all the inbound heavy cargo. But of course, what comes in must eventually go out. When it's time to take out the microscopic trash or secrete important proteins, the cell uses the exact reverse process, known as exocytosis. The cell packages the large cargo into a bubble, moves it to the border, and seamlessly fuses that bubble with the cell membrane, effectively spitting the contents back out into the outside world. As we wrap up this explainer, I really want you to remember that everything we just covered isn't just abstract science on a page. Your own body is made of these exact selectively permeable membranes. They are constantly eating through phagocytosis, drinking through pinocytosis, and managing heavy cargo just to keep you alive. Just think about that for a second. How many trillions of these incredible microscopic checkpoints are silently operating, shifting ions and macromolecules inside you right this very second?